Hi everybody, this is going to be a quick review of what I would study if I were you for your Biology 1 lab midterm. Alright, it's going to go something like this. If I were you, I would know about the different parts of the microscope. So microscopes are incredibly important in science. You need to know about the parts and pieces therein. Be able to tell me about everything top to bottom, be able to walk me through all the lenses, so 10 power oculars and all the objective lenses that we use, be able to tell me about total magnification, be able to tell me about working distance. Yeah, I think that'll do. Total magnification, working distance, that's perfect. Oh, and be able to walk me through how to set the scope up itself, so starting a low power, going up, tell me about when you'd use coarse and fine focus, all that fun stuff. Alright, you need to be able to understand how we use the metric system for just given measurements. So if I give you weights, you need to be able to convert that from common weights from one to the next. So, or alternatively like lengths. So if I give you millimeters, be able to convert that to centimeters, that sort of thing. Right? Just be able to just have a good grasp of what we would use for certain tasks. So if we're measuring very small amounts of water, you wouldn't be using liters, you'd be using milliliters. If I'm asking you how long it would take, or what unit of metric measure you'd use to travel from, I don't know, Shelton to the University of Alabama, be able to tell me that you'd use kilometers and not meters. Like, just a good grasp of the metric system and sort of what we'd use. Don't expect a pile of conversions on there. I don't find that to be particularly interesting, but there might be one or two. And just as we sit here, the test is going to be out of 50, and it's going to be fill in the blank. Okay. Let's see, what else do we have? Alright, the organisms from lab. So we did one lab where we went through and looked at different bacteria. We looked at different protists. We looked at different, like, some fungi. We looked at some animals. Just these different animals, or organisms I should say, and some of the unique structures that they presented. So you can guarantee that I'm going to throw a few of these on your exam and make you tell me sort of what's what. So be able to tell me like who the organism is it prokaryotic or eukaryotic, and then if there were unique structures like cilia or spore-forming structures or flagella, be able to tell me about that. And speaking of that, be able to walk me through the protists that we looked at, I should say, and tell me if they are ciliated, flagellated, or if they move via pseudopods, like a amoeba. Alright, we did several tests for, like, does it contain simple sugars or complex sugars? Does it contain lipids? Does it contain proteins? You need to be able to tell me what the names of those tests are for each one. So if I say, what would you use as a test to diagnose proteins? You better be able to tell me. And then you need to be able to tell me what a positive and a negative would look like for those tests. So you have a solution, you want to know if it contains starch, so you put iodine in it. If it turns very dark, that will be positive for iodine. If it stays like, I don't know, like tea colored, it would be a negative for iodine. So be able to tell me about the positives and negatives for these various tests. Okay, be able to measure volume of liquid in a graduated cylinder by reading meniscus and tell me what meniscus indicates. We looked at cell models, both plant and animal. Be able to tell me what these plant and animal parts internally are with the cells. So, for instance, chloroplast versus mitochondria. And speaking of chloroplast and mitochondria, be able to walk me through internally their structures, just sort of how they fit together. You have cellular respiration versus photosynthesis. What cells do cellular respiration, and which ones do photosynthesis? Be able to identify various pieces of lab equipment, like a scale, like a beaker, a flask, a graduated cylinder, yardstick for that, or a meter stick I should say for that matter. Okay, I will provide you with some graphs, and on those graphs you need to be able to sort of explain to me what you see. So if you recall back when we did tonicity, we talked about placing dialysis tubing bags in hypertonic, hypotonic, and isotonic solutions. I want to know what that would do to the weight of the bag, and if I present this to you in chart format, like on a table, you need to be able to read the table and understand what the changes in weight might mean in terms of isotonic, hypertonic, whatever. And again, all of that dialysis tubing stuff, that was just a good demonstration of passive transport mechanisms. And for that matter, like diffusion and osmosis, be able to tell me how you could influence rates of diffusion and osmosis. So what is it that influences how fast or slow things diffuse? And there's a lot of those that we gave. Your cell membranes are selectively permeable. You need to understand what that means. And tell me about the phospholipid bilayer. Okay, you need to be able to walk me through qualitative versus quantitative data. Be able to tell me about the scientific method. So if I present you with something, be able to tell me if it's a hypothesis or a conclusion or what have you. So qualitative and quantitative data, general terms for the scientific method, and let's see what else. Yeah, I think that's good enough. Alright, and just some general lab knowledge. So being able to read a thermometer, knowing which scale to use, understanding the difference between plant-eating animals versus animals which consume secondarily, those being autotrophs and heterotrophs. Yeah. And last but not least, you're a Homo sapien, and I need you to know how you might write your scientific name, or a scientific name for another organism like Panthera leo or something like that. What does Panthera mean? What does leo mean? These are genus and species. This is the Linnaean taxonomy system. It would not be the worst idea ever to be able to walk me from domains down to species. And I think that is that. I think that's good enough. So that's a good little review for you to help you do better on this lab test. And I think that's got it. Alright, thanks folks. Have a good day.